Um, Hi, thank you all for coming here. Pleased to welcome you to our Realizing Healthier Pregnancies Research Cluster. My name is Floor Spaans. I am the Research Associate and Lab Manager for Dr. Sandy Devich, and I'm pleased and honored to serve today as your session moderators for our presentations. On behalf of WCHRI, I would like to thank you for taking the time to attend our sessions. I just want to go through some acknowledgements before we get started. Let me share the slides. Bear with me. Right. How do I get this? Here. Okay, so first of all, the University of Alberta acknowledges that we are located on Treaty 6 territories and respect the histories, languages, and cultures of the First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. I also would like to, next, yes, I would like to thank WCHRI's funders, the Stollery Children's Hospital Foundation, as well as the Alberta Women's Health Foundation. And I will stop sharing. Okay. Just before we get started, WCHRI is actively seeking to build this research cluster. And so if you want to join this cluster or nominate any collaborators or know people that might be interested in that, please reach out to WCHRI. Michelle is here from WCHRI, who started this session, and she will stay on after the session to answer any questions you might have about participating in the cluster. As you may have seen, the session will be recorded and it will be posted on the WCHRI website, but please allow us 24 hours to access the session recording. And WCHRI will send out a link to everyone that was here. We're looking forward to sharing our research with you, and we encourage questions after each of the sessions. We have the two presentations. I'm pleased to introduce Dr. Meghan Riddell, as well as Dr. Helen Jones, to present to us today. Dr. Meghan Riddell is an assistant professor in the Department of Obstetrics and Gynecology, as well as the Department of Physiology here at the University of Alberta. Her research focuses on the formation of the placenta and the adaptation of the uterine lining during pregnancy. Her interest includes understanding how deficiencies in these processes can lead to the development of pregnancy complications. And then our other presenter, our second presenter today, is Dr. Helen Jones. She is an associate professor in the departments of physiology and aging, obstetrics and gynecology, as well as molecular genetics and microbiology at the University of Florida. And she is also the co-director of the Center of Research for Research in Perinatal Outcomes. Her research studies are focused on placental physiology and pathophysiology, placental vascularization and placental therapeutics, and the impact of these on lifelong health of both the mother as well as the offspring. Her research is funded by NIH as well as NICHD and the Wisconsin National Primate Center. All right, I would like to invite Meghan to present to us. You can share your slides.
Thank you, Flor. I will try. And there we go. We've got the right view. Great. Okay. Thank you very much for everybody being here today. So I know that this cluster is quite a diverse background, and of the names other than Samai Shahas, who's my PhD student in the audience, I did recognize that there's a fairly diverse background. So my apologies, I'm presenting mostly published data today, but some data out of the lab that I thought had the most interest for a group that's really interested in moving pregnancy field forward towards treatments. So I'll be talking about one of the first discoveries that we made when we started my research group. And I'm giving away the end of the story first, so you'll have to bear with me as we build back to it. That the cells in the human placenta undergo a form of cell death called pyroptosis. And the reason I have to give away the ending first is because we set out to discover something else entirely, and this is where we ended up. So this is a bit of a winding road of a presentation, but nevertheless, please come with me for the next hour. I suspect that Helen will be here as well, as we get to understand better the most magnificent organ that you no longer have, the placenta. So I adore this picture because it really highlights just how important the placenta is in pregnancy and to all of us being here and being healthy adults today. So the placenta forms at the very earliest stages of pregnancy and has to be completely functioning by the end of the first trimester. And the human placenta in particular, as you can see really nicely in this picture, is made up of many different finger-like hair projections known as villi, which are out in ultimately bathed in the blood of the maternal intervillous space, so the maternal blood. And to be able to understand placental physiology, it's best to understand really the placental structure. So moving into a cartoon view of really how the human placenta goes together. So you can see these many different finger-like villus projections. Here, the white space is the blood that's delivered through the uterine arteries, the maternal blood, and all of these placental villi are bathed in it. And one of the primary functions of the placenta is to facilitate maternal fetal oxygen, nutrient, and waste exchange. And you can appreciate that by floating in a pool of maternal blood, the placenta is ideally placed for this. And as this cartoon really nicely highlights, at the core of every one of these finger-like projections is the vasculature of the placenta. So ultimately, all of this placental vasculature comes together into the umbilical cord to deliver things back and forth to the fetus. So my research program is based in the human placenta, and the human placenta is essentially comprised of 4 different types of cells. So the stromal cell compartment, so these are different cell types that perform mechanical stability of the placenta, and are also important for setting up different stem cell niches. So the vascular cells, so the endothelial cells, paracytes, and smooth muscle cells that form the core of those vessels that really are at the heart of every one of these villi. Hofbauer cells, which is a placental-specific population of macrophages. And then the star of the show and my first love of cell types, the trophoblasts. So these are the epithelial cells of the placenta. So the epithelial cells of every tissue are those that form the outermost lining, so that are in contact with the environment. So in the placenta, these ones are on, as you can see here, the outside lining of the placenta and bathed in the maternal blood of the intervillous space. So in the placenta proper, there's 2 different types of trophoblasts. So the first is the cytotrophoblast, so this mononucleate population of cells, which are proliferative, so they can expand. And their purpose is ultimately to allow for the proper formation and function of the most bizarre cell type that a human ever has, the syncytial trophoblast. So this is a single gigantic multinucleate cell that covers the surface, the maternal-facing surface of the human placenta. And for some perspective, by the end of term, it's been estimated to be approximately 12 meters square and contain billions of nuclei. So critically, the syncytial trophoblast is a post-proliferative cell. So it can't replicate its own DNA, and it requires the incorporation of these cytotrophoblast progenitors in order to expand and regenerate and continue to function. So it's a balance between these 2 cells that really is required for the maintenance of this maternal-facing surface of the placenta. Well, the syncytial trophoblast is an incredibly important cell, as you can guess just by its location. But it really carries out many of the functions of the human placenta. I love to say single-handedly, though not single-handedly. So for example, it's a site of hormone production and secretion. So this is an image taken by my PhD student, Samaya, who's here 6 times. Of the production of the hormone human chorionic gonadotropin, which is the basis for the pregnancy test, which is produced by the syncytium. As I've already mentioned a couple of times, nutrient transport is critical, and this occurs primarily at the syncytial trophoblast due to the fact that its apical or its maternal facing surface is directly bathed in maternal blood, despite the cell being of fetal origin. So it's encoded by fetal DNA. And it also is incredibly important as both an immunologic and a barrier to infection. So which is really where we're going to stay today. Because it's this remarkable and very unique space where you have a fetally derived cell directly bathed in and directly interacting with maternal cells. And many of you are likely aware of the immune paradox of pregnancy, that the fetus is a semi-allogenic graft, basically. It's half not itself. And here it is, this one gigantic cell is responsible for making sure that the maternal immune system is not able to cross and invade. And in most situations, it's also involved in the dialogue between the maternal and the fetal immune system to make sure that there's an adequate balance. But it's also a highly effective barrier against pathogens as a result, because of lacking the normal cellular borders that occur between epithelial cells. There's not the space for different pathogens to break through and enter into the fetal compartment. So it's really incredibly well adapted and has evolved in order to be highly resistant to pathogen entry. And it plays a really important part in the inflammatory balance of pregnancy and in this defense against pathogens by releasing different factors such as antimicrobial peptides, cytokines, which prime the maternal immune system, and different pieces of material and lipid encased vesicles like exosomes in order to help this really effective barrier be maintained. And it's also capable of carrying out innate immune responses. So a reminder that the innate immune system is that first line of defense that then awakens the adaptive immune system. And when the few pathogens that can infect the placenta do infect the placenta, it can mount these innate responses. So that's this dialogue between the mother and the fetus is incredibly important. And therefore, it's been shown that placental cells can actually mount inflammatory responses to multiple different conditions. So the one where you would expect it the most is the rare cases of the different organisms that can actually infect the placenta and cause congenital infection of the fetus in utero. So, for example, Listeria monocytogenes or Zika virus elicit an inflammatory response from the cells of the placenta. As well, the placenta will mount inflammatory responses during times of maternal infection. So in the absence of the infection of the placenta itself, in chronic maternal inflammatory disorders, such as autoimmune disorders, and it will also mount inflammatory responses to damage-associated molecular patterns like uric acid, which are released from the maternal compartment. So it's really responsible for this dialogue between the mother and the fetus, and inflammation is a key output in order to reattain homeostasis. So that's really the point I wanted to remind everyone next, is that inflammation is actually a normal and a healthy balance and absolutely critical function for a tissue. It's when there is an excessive or a failure to resolve inflammation that things can really go wrong, and we start to develop inflammatory diseases, which also occur in the placenta. But under healthy conditions, we have healthy inflammation, which allows for the mounting of an immune response or the clearing of a damaged tissue in order to return to homeostasis. But when things go wrong, there are profound impacts in pregnancy for all of the players at that maternal fetal interface. So inflammation and infection have really severe consequences in the maternal compartment in terms of an increase in the inflammatory response to certain initiating factors during pregnancy. There can be sepsis or obstetrical hemorrhage. When there's an imbalance and an increase in inflammatory mediators, some individuals will go on to develop dangerous pregnancy complications such as preeclampsia, which is the de novo onset of hypertension in pregnancy. At the placental level, chronic inflammation or excessive inflammation will lead to decreased nutrient and oxygen transport. And the oxygen and nutrients are needed for the placenta itself, not just for the fetus. It can also serve as a reservoir for reinfection of different types of pathogens. And placental inflammation can manifest as villitis, so the villi of the placenta can become inflamed, and there can be invasion of maternal white blood cells as well as fetal. And this can lead as well to the deposition of fibrin within the placenta, which harms it even further. And ultimately, this inflammation can have impacts on the fetus. So placental-derived cytokines can be delivered into the fetal compartment and lead to microcephaly and other neurological developmental disorders, growth restrictions, so the impairment of the growth of the fetus, which is associated with a lifelong increased risk of cardiovascular disease. So the placenta is right at the heart of all of these different impacts for two individuals that can really be long-lasting. And one of the important features of inflammation is that it's inextricably linked to cell death cascades. See, I told you we'd get back to pyrotosis eventually. We're just a bit of a winding road here. So the more famous forms of cell death are most studied forms of cell death in all of biology, these non-inflammatory apoptosis. So this is at its core, it doesn't cause inflammation due to the orderly disassembly of the cell. But there is also an increasing number and an increasing frequency of knowledge that there are highly pro-inflammatory forms of cell death that are also strongly controlled and turned on under specific situations. So in this pro-inflammatory arm of cell death, for example, we have necrotosis and pyrotosis. And the characteristics of these cell death pathways is really that there's an increase in the membrane permeability, a release of damage-associated molecular patterns and cytokines, and ultimately, they're highly pro-inflammatory, so they elicit an immune response. So diving a little bit further into these forms of cell death, because they're much more recently discovered. So the key feature of both necrotosis and pyrotosis is the formation of pores in the plasma membrane of a cell. So it's quite a busy slide, but ultimately the effectors is this pore formation that allows for the permeability and the release of the different factors that are pro-inflammatory in these forms of cell death. So for necrotosis, this is ultimately the formation of pores in the mixed lineage kinase domain-like family. And for pyrotosis, the membrane pores are formed by gasdermin proteins. And these are tightly regulated processes in order to not have their faulty turning on, and then you have cell death and inflammation when you don't want it. Now, in the placenta, when I started this project, what was known is that necrotosis can occur in the trophoblasts. And so it's another important thing to appreciate is that though immune responses are carried out by immune cells, inflammation, it takes the dialogue between multiple different cell types, and particularly epithelial cells like the trophoblasts play a key role in the amplification of these pathways. And so when we started this work, we knew that necrotosis can occur in the placenta as well, so these inflammatory responses, but Isabella Canegia's group at the U of T had shown that it was occurring in the cytotrophoblasts, so those progenitor cells, and not in the syncytial trophoblast that's actually bathed in the maternal blood. So that's kind of where our story about necrotosis ends. But in terms of pyrotosis, just for a little bit more background before we take a sharp right turn away from pyrotosis so that I can show you how we came about this discovery. There's also additional complexity because there's multiple different types of gasdermin that can actually carry out this form of cell death. So the most studied type of pyrotosis is carried out by the gasdermin D family. not family, the gasdermin D protein. So this has been identified about 10 years ago, that gasdermin D is activated downstream by proteolytic cleavage by caspase 1, and that it's activated in particular downstream of the NLRP3 inflammasome to form these pores in the plasma membrane, releasing cytokines like IL-1 beta, and under certain conditions can lead to cell lysis. But another family member that has also been shown to elicit similar responses in cells is gasdermin E. So highly related to gasdermin D, but activated through a different mechanism by different proteases, and independent from the inflammasome activation, with the same outcome of leading to these pores in the plasma membrane. So as you can guess, this is what my lab discovered, that the syncytial trophoblast undergoes pyrotosis, as I've told you 6 times already. But how did we get there? So we got there when we were actually attempting to understand a completely different, very important part of a cell's homeostatic regulatory mechanism, cell polarity. So I told you the hard right-hand turn. So cell polarity is the asymmetric distribution of biomolecules within a cell. So it can be characterized by the secretion or the laying down of different molecules from the top to the bottom of the cell. So in a blood vessel, for example, they lay down collagen 4 sleeves on the outside of the vessel, but will release protocoaxin into the interior of the tube. It's a highly conserved hallmark of epithelial cells like trophoblasts, where they have the accumulation of different biomolecules at their apical surface than their basolateral surface. And one of the key things that, though this is a fundamental characteristic of particularly epithelial cells that we even teach quite early in our education system, that even though it's this hallmark, that it has to be actively maintained. So polarity is maintained through the reciprocally antagonistic actions of kinases that localize to different areas of the cell. And when I started my lab, I was interested in understanding how the syncytial trophoblast is polarized, because it's an epithelial cell. It is magnificently polarized, as I'll show you in a couple of slides. And we started to look into a family of proteins known as the PAR polarity complex. So this is a complex of proteins that's evolutionarily conserved from worms through to mammals to regulate apical basal cell polarity. And it's comprised of 3 different family members, so partitioning defective 6 and partitioning defective 3, which are scaffolding proteins, which ultimately localize and activate the functional member of the complex, atypical protein kinase C. And just to make things confusing, there's multiple types of APKC, but that's all you need to know, because basically for this presentation, I'm referring to them as a group for simplicity's sake. But we wanted to examine whether APKCs, both types of APKCs, iota and zeta, may regulate syncytial trophoblast apical basal polarity. And so in order to look at this hypothesis, we were taking advantage of the fact that the syncytium is highly polarized. So it's actually a very easy unit to measure polarity in the syncytial trophoblast, because their apical surface is highly microvilliated. So the placenta itself is villi, and then on top of that, what we have at the surface of the syncytial trophoblast is more finger-like projections. But this time, it's not cells, it's actually just membrane, membrane filled with actin. And you can appreciate from this transmission electron micrograph just how heavily microvilliated this maternal blood-facing surface of the syncytium is. And this scanning electron micrograph really shows you just the complexity of that maternal-facing surface of the syncytium. So to be able to examine whether APKCs may be regulating polarity, we wanted to use these microvilli as a unit, because they have to be actively maintained through polarity regulating pathways. So what we did was we used a floating placental explant culture. So this is intact pieces, tiny pieces of the placenta that are cut, trimmed, and then put in culture medium. And we treated these with a total APKC inhibitor, and then prepared the samples for scanning and transmission electron microscopy. And this is when our story started to be less about polarity and more about us going, uh-oh, something is really wrong with these cells. In our transmission electron micrograph, you can see in the control treated explants on the left that there was nice microvilli on this maternal blood facing surface. And with APKC inhibitor treatment, polarity was definitely disrupted. There was a lack of microvilli on this maternal facing surface, but this cell is really looking like it's having a pretty bad day. There's a profound decrease in cytoplasmic density. These black round dots are mitochondria, which seem to be falling out of the cell. So it was our first indication that we were touching on something that was bigger than just the repositioning of proteins within the cell. And when we looked at our scanning electron micrographs, it got even worse. So on the left, again, the control, you can see the beautiful smooth membrane with the in between the membrane projections of these microvilli. With the APKC inhibitor treated membrane, you can see an almost lace-like appearance to the surface and the appearance of apparent pores into the cell. So we wanted to address that exactly. Was there an increase in the permeability of the syncytial trophoblast? Were these truly holes in the plasma membrane? And so at the time, we had a very talented undergraduate student named Jasmine Nugent working on the project. And so she developed a method for testing the permeability of the syncytial trophoblast. And to do this, she again used human placental explants and APKC inhibitor treatment. And then she added fluorescently labeled dextran, that's 10,000 Dalton molecular weight to the explants, and then wash them extensively with the idea that the dextran would only enter into the syncytial trophoblast regions that were permeabilized or be taken up by macropinocytosis, which would show up as very small punctate dots. And then all of this was imaged via whole mount staining by confocal microscopy. So we took advantage of whole mount staining and confocal microscopy and the structure of F-actin in the syncytium in order to identify the position of the syncytial trophoblast. So you can see here the magenta on this apical surface is beautifully, I call it snowflaked, that is showing where the different microvilli are, where that underlying mononucleate progenitor has a much more smooth actin cytoskeleton. So you can see the position of the syncytium this way. So using these methods, what Jasmine saw is what we would expect from the scanning electron micrographs, that the APKC inhibitor treated tissue, there was a regionalized uptake of dextran within the syncytial trophoblast that really filled the cytoplasm, versus in our control tissue where we saw the punctate high fluorescence dots consistent with macropinocytosis or cell drinking. And this was significantly increased the dextran uptake with APKC inhibitor treatment. So then that's when we went to the literature, learned that Isabella Canidia had already shut the door on necrotosis, so we thought we should really look into pyrotosis. And so we started first with the canonical pathway of gastromin D. So Christina Megli and Carolyn Coyne had shown that there's really extensive expression of gastromin D within the syncytial trophoblast already. But when we used an antibody to stain our tissue that identifies the active form of gastromin D here in magenta, we couldn't see an appreciable signal in the overlying syncytial trophoblast. So it didn't appear to be gastromin D pores that were forming. So we turned to gastromin E because there was documented high levels of expression of gastromin E in the placenta. And that is where we saw, so this is using an antibody that recognizes total gastromin E, so cleaved or otherwise, And we could see that normally it fills the cytoplasm of the syncytial trophoblast with APKC inhibitor treatment. It is translocated primarily to the membrane and forms dots. And in order to prove that it was gastromin E that was allowing for the increased permeability, we again did the floating placental explant culture and combined it with siRNA-mediated knockdown, and again performed dextran uptake assays. And as we expected, there was a complete loss of the APKC inhibitor treated increase in dextran uptake with gastromin E knockdown. And in order to say that the permeability was bidirectional, not just with the dextran getting into the syncytial trophoblast, but that there were factors that were also being released through, most likely through these pores. We also looked at the release of IL-1 beta from our explants, and we saw there was an increase in IL-1 beta release with APKC inhibitor treatment, as well as an increased release of human chorionic gonadotropin, which is produced in the syncytium. So, in summary, APKC inhibitor treatment leads to profound alterations in the structure of the syncytial trophoblast and the gastromin E-dependent pyrotosis. But this isn't particularly physiologic. So we then wanted to understand whether there was a physiologic stimuli that would be able to elicit pyrotosis. And I have gone much too slowly, so I'm going to have to go a little bit too fast. Michelle told me to be comfortable and relaxed here, so I've gotten too relaxed. So my apologies, we're going a little faster now. But and from the literature, what we could find is that TNF-alpha, so an important pro-inflammatory cytokine, is known to decrease the expression of APKCs in intestinal epithelial cells. So we thought that we would see whether TNF-alpha had the same effect in the placenta. So we did show that in our paper that TNF-alpha decreases APKC expression in vitro differentiated syncytial trophoblasts. And then, as you would expect, since it decreases APKC expression, TNF-alpha also led to the same sorts of effects that we saw with APKC inhibitor treatment in both term and first trimester placental explants. So scanning electron micrographs showed the formation of these pore-like structure and the regionalized loss of microvilli. It also increases permeability. So this is at doses of TNF-alpha that are actually seen in maternal circulation during preeclampsia. So an established inflammatory can be an inflammatory syndrome of pregnancy. And gastromin E could be localized to the apical membrane, and treatment of in vitro differentiated syncytial trophoblasts led to a twofold increase in this cleaved form of gastromin E. So yes, gastromin E is still working. And knockdown of gastromin E also blocked the effective TNF-alpha on syncytial trophoblast-induced permeability. And then the last thing that we wanted to see was whether there was a release of these membrane-bound vesicles and whether we could detect whether there was cell lysis in the syncytial trophoblast. So we used a live cell imaging system that we had recently acquired in the lab and performed serial imaging every 30 minutes of placental explants. So just to remind you that the surface of this is, even though we don't have any labels for the syncytial trophoblast, the surface of the placenta is formed by the syncytium. And so when we captured images, we can see here, watch here, really after 17 hours, the membrane, the surface of the placenta really begins to almost boil and bubble. So the initiation of pyrotosis is also leading to the release of large membrane-bound vesicles into what would be the maternal blood. So ultimately, what this work has shown is that the syncytial trophoblast can undergo this highly pro-inflammatory form of cell death, and that it releases pro-inflammatory cytokines like IL-1 beta, and this would be directly into the maternal blood of the intervillous space. And that is a very high risk kind of procedure to do, to be popping holes in yourself when you aren't even actually that person, and you're bathed in someone else's blood. but that's really what is occurring. And so there's many unanswered questions really stemming from this work. Because as I highlighted earlier in the presentation, the initiation of the inflammation is normal and can allow for the reattainment of homeostasis. But when it goes awry, it can lead to pathologies. So one of our most important questions is, does the chronic or excessive initiation of syncytial trophoblast pyroptosis lead to placental dysfunction? So can this actually be one of the causes for placental malfunction and inflammation? And then the second most important question that we're trying to answer now is, what other factors may actually initiate syncytial trophoblast pyroptosis? Because TNF-alpha levels can go up under many different situations. And is there other initiating pro-inflammatory factors, or what other factors will actually cause this really dramatic event to occur? And the reason that we're pursuing this is because with the identification of pyroptosis, there's also been the identification of already in use pharmacologic agents that can block pyroptosis and have been shown to be effective in treating pro-inflammatory disorders like remitting and relapsing multiple sclerosis. So one of these is called dimethyl fumarate, and it blocks gastromin D and gastromin E pore formation. And we have shown that incubating placental explants with this drug, which is already used in the adult population, can block syncytial trophoblast pyroptosis. So having the opportunity of a cell that's right at the maternal interface and bathed in the blood, we feel it's a prime target for treatment. And if we can repurpose or use, I'm sure Helen will talk about her magnificent drug delivery systems in her talk, to be able to get antipyroptotic treatments to the syncytium, could we have the opportunity to block placental inflammation. But I just wanted to finish up with a word of warning that, as always, we have to understand much, much more. And especially when it comes to the syncytial trophoblast, I'm not sure how many people in the audience work in it, but this cell just marches to the beat of its own drum. It takes everything that everybody else has and says, I'm not going to do it that way. I'm going to do it my own way. And we have a bit of data showing some pretty paradoxical things that are occurring in the syncytium. So when we started to examine the role of gastromin D in the syncytial trophoblast, so reminding you, this is the canonical mediator of cell death in most cell populations. And when we knock down gastromin D with siRNA in the syncytial trophoblast, it actually induces death. So that's the exact opposite of what you would expect. So the loss of gastromin D induced permeability to dextran in the syncytial trophoblast. And when we add TNF-alpha in addition to gastromin D knockdown, things get massively worse. So TNF-alpha leads to this characteristic regionalized permeability of the syncytium to factors, which we believe is really important for making sure that the cell can survive. But when you knock down gastromin D, you can see that there's really an increased permeability and much more. And it actually amplifies the cleavage of gastromin E. So the syncytium is working exactly opposite to everybody else. And here, gastromin D is a pro-life, pro-survival signal, and we're still identifying the mechanisms that works through. So there's much more to be learned in order to make this an effective target. And with that, I'd like to thank you all for my right-hand turns and twisty-turvy road. It's a very good lesson of keeping an open mind when you're looking at a project, because you start one place and end up another. And just thanking everyone in my lab who has contributed to the work over the years and continues to contribute to the work, especially Kashali and Jasmine, Wendy, Samaya, of course, Ivan, who's holding down the fort on pyroptosis still, all of our funding agencies, and all of our tissue donors and our research nurses who help us to get placentas. I'm happy to take any questions.
Thank you, Meghan. Well, that was amazing and scary. And so Yes, anybody who has any questions, please use the raise hand feature, and I'll do my best to invite you to voice your question in about the order that the hands are raised. If you're more comfortable using the chat feature, you can also ask your question there, and I will do my best to voice your question for you. But please let me know if I'm doing anything wrong. So yeah, looking forward to questions. Any questions for Meghan? Don't be shy.
I also went too long, so.
That's okay. We got a lot of maybe I'll start with a question if you don't mind. It's just more out of curiosity. So you're seeing this pyroptosis in the syncytial trophoblast and then the necroptosis, the other one, in the cytotrophoblast and not in the syncytial trophoblast. Do you have, like, I don't really know the differences between these two, but do you have any thoughts on why this is so different and like what might be, why not necroptosis in the syncytial trophoblast?
I'm the necrotosis pathway, that's work from Isabella's lab, and it seems like there's not a high expression of that mediator that's required for forming pores from that pathway in the syncytium. Although I'm not sure, other people are looking into it more. It's really highly likely that there's multiple pathways that can be executed by the syncytial trophoblast. That it's not just pyroptosis, how it dies. We know there's lots and lots of work from the past that apoptosis doesn't occur in the syncytium anyway. And one of the things that I anticipate is that when we go in and we block pyroptosis, that it may actually activate other cell death pathways. So when something wants to die, there's many different ways to do it. But there's something inherently different about the syncytium, probably because of its structure, that really, it does make it harder. That's why we were so surprised, because it is harder to die when you're a gigantic cell. I don't know.
I think that's good. Sylvie Gerard.
Yeah, hi, Megan. That was a great talk. I just want to get your opinion because you've shown beautifully that these cells, like the trophoblast, is polarized. And I'm just wondering, when they die and they release mediator, you talked a lot about release in the maternal circulation. Do they release anything on the other side as well, and then can have impact maybe on Bauer cells or in any? And in your beautiful explants model, did you see anything perhaps on the other like cells that are inside the villi?
We haven't looked extensively enough. The problem with the explant system is that the villus core is starting to die all on its own. So it's more difficult to do it. But I would really love to develop a system with just like primary ST differentiated on a transwell, so that then we could see when we were inducing pyroptosis what was released into the maternal versus the fetal compartment. Because I know that there's work that has shown, for example, Zika virus infection in the placenta leads to the release of IL-1 beta. And our data would suggest that that would be from a combination of the syncytial trophoblast and probably the Hofbauer cells. And that IL-1 beta ultimately has really negative effects on the fetal brain. So I think that it's probably going both directions. I mean, the coolest part of the data that I didn't get to show today is that depending on the gestational age, so term placental explants, we never see an obvious signal for gastromin E in the apical surface of the syncytium, so the blood-facing surface. And there actually isn't an increase in the release of IL-1 beta in those cells. And we don't know if that's because there's less IL-1 beta that is actually made in the syncytium at the end of gestation, or whether it almost looks like the IL-1 beta may be being released in the other direction. The pores, there's permeability, but there seems to be more pores that look like they're forming with the fluorescent staining anyway on the basal side. So I think it probably goes both ways, but we don't have any data yet.
Thank you.
Yep.
No more questions? Just one quick question, Megan. Just thinking about the TNF alpha dose and linking it back to preeclampsia and increased TNF alpha levels, for example, in the mother, which I think has been shown. So how does that relate to the doses that you used? And I mean,
Yep,
obviously what you're seeing is very serious.
Yeah, no, the TNF-alpha doses that we used are actually from the literature. So they're equivalent to the concentration of TNF-alpha in the maternal circulation during preeclampsia. So
That's scary.
yep, isn't it? Yeah.
I mean, it must have some mechanism to protect itself a little bit from this, because it sounds like an how do you say, point of no return when you go into this.
Yeah, well, that's it. So we're looking into, because of it being a single giant cell, that there must be repair mechanisms and like and pro-survival signals that have to be initiated at the same time as the death in order to keep the balance.
Yeah.
So, and that's what we think, is that then there may be the opportunity for, if it goes on too long or there's too much pyrotosis, that then it can spread. And you can see, I mean, if this spread too widely, it could just lead to miscarriage. So undoubtedly, it's so high risk, it has to be highly regulated to be able to turn it off. So, but we have to develop the systems to be able to look at the timing and how it would be able to be recovered and that kind of stuff, because they're more complicated experiments. But we're trying. Ivan is trying very hard.
Do you think that your results would be different in like a term placenta versus the early placentas you're using? Or?
Yeah, we did it in both. Actually, I just didn't really get to show it today. And so there are differences between early and late gestation. So like the IL-1 beta release, the pores still form. So it may be that the outcome of pyrotosis is different based on the gestational stage as well, which is also showing that it may be a fairly common event if there is differences that are observable kind of right away. That it's something that is likely a form of dialogue between the syncytial trophoblast and the placenta and the mother, or as well between the fetus and the placenta.
Okay. Any more questions? If not, that's okay, because we do have time after the presentations to answer some more questions if you want. Speakers will stay along, but since we are running a little bit late, I will move on to Dr. Helen Jones. Yeah.
Probably see far too much right now. Okay,
Yes.
can you see the right one?
Looks good.
Oh, right. Can you still see the right one? Okay, good. Okay. You're looking at right.
Sorry, Helen, we're now seeing your presentation. Yeah, the other one. Yes, that's right. Yeah, perfect.
Okay, so thank you very much for the opportunity to come and tell you all about developing placenta therapeutics today. Thanks, Meghan. I don't have to do anything on placenta, so that's marvelous. You now know everything about placentas you could ever need to know. So that helps. So I'm going to tell you a little bit about our story over the last 15 years and how we've been developing placenta therapeutics. We've gone after the low-hanging fruit of fetal growth restriction, but our delivery mechanism can be used probably for many, many different types of placental complications. So very briefly, because I know some of you on here have no idea what fetal growth restriction is. It's a problem that occurs around 10% of all live births within the US and Canada. But if you go away to other places around the world, sub-Saharan Africa, for example, it occurs in about a quarter of all live births. So it really is a significant impact on global population. It can occur in isolation, or you might have seen on Meghan's slides, it can occur with preeclampsia, also genetic syndromes and congenital heart defects as well. It results in iatrogenic premature delivery. What does that mean? That means the neonatologist has had enough of waiting for that fetus to start growing properly, and they want to deliver it and get it into the NICU. And of course, ending up at the NICU at any point during fetal life is not really the best case scenario, despite many of the advances of the neonatologists. It not only impacts those very small neonates, they then carry with them lifelong sequelae, which can include things like cardiovascular disease, obesity, type 2 diabetes. And in fact, if we could figure out how to fix fetal growth restriction, we'd have a much healthier global population to start with. About 70–75% of all cases of growth restriction have some underlying placental issue. So that makes it an opportune place to actually think about therapeutics. But when we think about therapeutic interventions during pregnancy, of course, we've got two individuals, a maternal-fetal interface, and those two individuals want to go on to have healthy lifespans for the rest of their lives as well. Currently, like I said, early delivery and NICU admission. But maybe looking to the future, could we improve the placenta, improve the placental function, and does that improve fetal growth? So why are we choosing to target the placenta? Well, the placenta is the organ that doesn't get taken home at the end of the day, generally gets thrown in the trash. Everybody wants the thing from the other end of the umbilical cord. The whole of that surface area towards delivery, so what was that, 12 meters squared? It's about half a tennis court, if you need to visualize that. That's actually accessible via the maternal circulation. So if you can get something into the maternal circulation, you can get it to all of that surface area of that syncytial trophoblast. And of course, generally it's discarded after birth or handed to someone like myself or Meghan waiting outside labor and delivery. But again, therapeutics in pregnancy comes with a lot of problems and a lot of challenges. It needs to be targeted. We don't want to treat all the cell types. It needs to be specific. We really want to go after one particular pathway, for example, rather than having to treat 4 or 5. It has to be highly effective and efficacious, and it needs to be safe. Again, both of these individuals want to go off and live long, happy lives after this pregnancy. So about 15 years ago, I started working with a group at the University of Cincinnati with a polymer chemist who let us have access to this copolymer. And I am not the polymer chemist, but I can tell you a little bit about this copolymer. It's non-viral, which is very important for pregnancy, of course, to start with. It's also currently this copolymer has been used in multiple different types of makeup preparations. It has FDA approval for external skin use, and so it's not seen as a toxic moiety once it's formed its particle. It has 2 sections to it. One is to condense the DNA that we're delivering, or the plasmid that we're delivering, and the other end is to avoid recognition by the immune system. And we've never seen any evidence across the years of any recognition by the maternal immune system across multiple species. Inside of our particle, we like to deliver DNA plasmids for overexpression, but of course we could deliver anything within that particle. Our particle forms based on charge. It's extremely easy. It assembles by itself. In fact, the most complex thing you have to do is put it in a tube and go and have a cup of coffee. The DNA plasmid itself is in here, protected away from the circulation. And again, this is the part of the polymer that cannot be recognized by the immune system. And we published that now just about 10 years ago. And we've done an awful lot with this polymer over the last 10 years. We've used multiple species, including mice, human ex vivo and in vitro studies, and guinea pigs. And we've demonstrated quite convincingly that our particle will not cross the placenta when delivered, either by direct injection or into the maternal circulation. It has a significant effect on the interhemal distance. That's the distance and the thickness of tissue between the maternal circulation and the fetal circulation. So for anybody that knows Fick's principle, that means that oxygen would be able to diffuse more easily across that smaller tissue barrier. In the short term, we see an increase in placental nutrient transporter expression across both mice and guinea pigs, and in the in vitro treatment of syncytia trophoblast. And in these FGR fetuses with short-term treatments, we see an increase in glucose concentrations. Now, of course, we don't really want to treat mice and guinea pigs. We're really interested in helping humans. So we wanted to know if our particle would be taken up into the human placenta. And so we collaborated with a group at Texas Tech who do fresh human placenta cotyledon reperfusions. So this is a piece of human placenta here within the system. On the right-hand side would be the red blood, which is meant to be mum circulation, and the left-hand side is the fetal circulation. And so these vessels at the top and the bottom of the cotyledon are cannulated, and flow is established, and it's a continuous flow that recirculates, and maternal and fetal circulation. So we set this up, and they were perfused for about 2 to 2.5 hours. Obviously, that's not going to be enough time for DNA to express, but we were able to label our particle, and so we're able to see our particle. So our particle has Texas red on the outside of it, so it fluoresces red. You can shine the right colored UV light on it. That was added to the maternal circulation and left to perfuse. Somewhere between an hour and an hour and a half. We wanted to give it a chance to be taken up, but we didn't really want to go beyond the lifespan of these fresh cotyledons. So what happened? You can see here in the maternal perfusate, before we added the particles, there was no Texas red readout, as you'd expect. We then added the particles, and there it is. You can see it circulating in the maternal perfusate. But by the end of the perfusion, that perfusate was actually reduced in the amount of signal that we saw. So it had clearly gone somewhere, and we wanted to know where it had gone. Where it hadn't gone is across into the fetal circulation at all. There was no evidence of it actually crossing the placenta and getting all the way through to the fetus. So it's no longer in the maternal perfusate, and it's no longer in the fetal perfusate. So where is it? Well, thankfully, Texas red you can see, and you'll appreciate here this central lower panel is a perfused human placenta, and here we have beautiful Texas red staining all around the syncytium. Okay, and this is taken up by the syncytium itself. Please excuse the occasional red blood cell which likes to get trapped, which really is annoying for our Texas red imaging. But you'll appreciate over here, you can see the nuclei of the syncytium around the outside of these villi, and then the syncytium stained red with that Texas red. And if you note over on the right-hand side, you'll see that even the numbers tell us that the placenta is full of that Texas red particle. So that was good. We knew eventually if we ever got to humans that the particle would get taken up by the placenta if it was in the maternal blood. So what do we do next? Well, we want to use a model to help us understand how we would actually treat growth restriction. And in the human currently, you can't predict growth restriction. You can only diagnose it following assessment of in utero growth. And so we really needed to utilize a model that had existing fetal growth. We'd use the mouse, but we had to treat at the same time as we actually interfered with the growth due to the length of the mouse pregnancy. So we decided to move on to the guinea pig for a couple of reasons. One being pregnancy length. Two, they're extremely relaxed animals, especially in Florida. You can see this one here hanging out in our vivarium. We look after them very nicely. They get to hang by the pool most of the time. And what happens with those guinea pigs is they're actually pregnant for almost 70 days. And there's a model that exists that has been published very well over the last 20 years that allows for induction of growth restriction by mid-pregnancy, which then gives us 35 to 40 days to actually treat these animals. The other great thing about the guinea pig is it's actually got one of the closest types of placenta to the human. It's much more similar to the human placenta than the mouse or the rat or the rabbit, and in fact, even compared to some non-human primates. So here's the model that had been designed for inducing growth restriction. Diet is restricted by just 30% for a month before pregnancy, and then the first month of pregnancy. And then at mid-pregnancy, they're actually transferred to a 90% food intake. Otherwise, we get massive fetal loss, and that obviously doesn't allow us to model growth restriction. We initially in our short-term treatment injected then at day 35. And here you can see our mother guinea pig finding out that she is indeed expecting. And she is pregnant at gestational day 20, which gives us 15 days to figure out how we're doing the treatments and what day of the week it is. And then we go ahead, anesthetize the animal and give the injection. At mid-pregnancy, there's no adverse impact on placental or fetal development giving our treatment. Visually and structurally, there's no different to the guinea pig placenta. This is a guinea pig placenta. This is the exchange region here, so very similar to the human placenta. This is a subplacenta and the interaction with the decidua, which again is more like the cell columns and the decidua in the human placenta. So no real disruption with that short-term treatment. And what we see in our model is we see that true growth restriction. We get a restriction of about 20% of fetal growth. And unsurprisingly, after just 5 days of treatment, our nanoparticle treatment doesn't do anything. We didn't really expect it to. What we wanted to make sure was that it wasn't making the situation actually any worse. The same with the placental weight here. It's not really any particular different from just the restricted animal here. We, of course, do everything in control animals as well with our treatment. So the control animals get nanoparticle as well to make sure that we don't get accidental fetal overgrowth, for example. We wouldn't want to actually grow too big a baby. That would be at the opposite end of the spectrum and not really beneficial for those patients. With that 5-day treatment, we didn't see too much change to those placentas, but we did see some changes. We actually saw a change in the interhemal distance of the structure of that placenta. So we're starting to see very subtle changes in the structures of those placentas, which were indicating that maybe we'd be getting better transfer of substrates like oxygen. We also looked at mTOR signaling. We're interested in nutrient transfer to the fetus. And really the mechanism for how the placenta is responding to that maternal reduction in nutrients. And so, although we did see some indicator of IGF maybe impacting the subunits of mTOR, what we actually found more interesting and more significant, in fact, was a de-repression of the DEPTOR molecule. So DEPTOR represses the mTOR signaling, reduces trophic effects on the placental cells. And in fact, in our restricted model, we see an increase in that DEPTOR compared to control. We see an increase in DEPTOR when we treat our controls with a nanoparticle. Again, this would be repressing any signal for overgrowth. So exactly what we would like, beautiful homeostatic mechanisms there being induced. But over here in our restricted animals that get our nanoparticle treatment, we see a de-repression. And so the repression of the DEPTOR is lifted, allowing more signaling, and then that would actually trigger more transfer of nutrients. So some very promising data from those mid-pregnancy studies. So the short-term treatment improves the placental signaling. It looks like it might help the diffusion of the oxygen as well as the transfer of the glucose. And it looks like we might be improving the in utero environment. And so we were really interested in, did that really impact fetal growth? So to do this, we wanted to expand our model out, and we wanted to go all the way to term to see if we could impact fetal growth at term. And this time, to ensure that we had constant presence of the extra IGF-1 we're delivering, we gave 3 injections across this latter half of pregnancy 8 days apart. And then we delivered them by C-section at day 60. As you can see here very clearly, the growth of the fetus is in fact restored to normal. This is really exciting. Our lab were literally doing backflips when we got this data through. And again, this is the growth restricted group here. So we have a complete restoration of fetal growth in these pups that were confirmed growth restriction at mid-pregnancy by ultrasound. Now, what's interesting is we didn't only have that restoration of fetal growth, we also have a restoration of fetal stress. So we know our fetuses have elevated cortisol. We know growth-restricted fetuses in the human, as well as in the models, have elevated cortisol levels and experience large amounts of fetal stress. And in fact, these cortisol levels can be blamed for future programming of the HPA axis and those problems with long-term impact being growth restricted. And so you can see beautifully here a complete restoration of these levels of cortisol back to normal following our treatments. And how is that happening? Well, our placentas are doing exactly what they should. The efficiency of the placenta is normalized. So the gram of fetus that you can grow per gram of placenta, again, has gone back to normal following these treatments. So we've really normalized the placenta, allowing for restoration of normal fetal growth and development. Now, as anybody who knows about placentas, male and female placentas are not the same. And so we always like to look at the impact on our males versus our females. And here you'll see we have difference in fetal blood glucose levels with our males, but not our females. We also see that at an intracellular signaling level within the placenta. So here we have the ERK kinase signaling pathway, showing again restoration within the females, but completely different changes to this pathway within the male growth restricted, and then no change with the treatments there in the males. And so we've seen that now with a few different signaling pathways in the placenta. There are 3 publications on their way out, currently under review, that have all of those signaling differences in, showing that the treatment does work in both fetal sexes, but the mechanisms that are disrupted in the growth-restricted placenta and those that are restored by the treatment are actually different between the males and the females. So that's awesome. If you're a growth-restricted fetus, we can fix you, we've got you. We know what we're doing, especially if you're a guinea pig. It's great. We have no more growth-restricted guinea pigs. But what about mum? Remember, there are 2 individuals in these pregnancies. And maternal stress levels in women that are diagnosed with any pregnancy complication are what should be obviously higher than a normal pregnancy. But in growth restriction especially, there's an association with elevated maternal stress in late pregnancy. And so we were interested if our treatment would impact mum. Obviously, we don't want any detrimental impact, but if there was anything that would actually be useful for mum as well. And so when we look at general physiology of mum, we see no abnormalities or changes with her blood glucose or her CBCs, for example. However, excitingly, what we do see is, once again, maternal blood cortisol is actually now reduced in these mums that were carrying fetuses whose placentas were treated with the IGF-1. The IGF-1 is likely not having any direct effect on the maternal organs, given that IGF-1 receptor is actually downregulated in the pregnant individual. And so this is likely secondary to a more normally acting and signaling placenta, reducing that maternal cortisol. So again, that's awesome. You're a guinea pig, that's great. But how do we get to the human? Well, to get to the human, we have to go via the non-human primate. And so that's currently what we're doing. We've got 2 new publications just coming out now, showing our prelim data and our initial studies with the rhesus macaque in collaboration with Jenna Schmidt up at Wisconsin National Primate Center. We have been using, again, a transgene plasmid complex with this copolymer. We give this at about gestational day 100, which would be the equivalent to the time in human when you would be diagnosing that fetal growth restriction. And we've left these individuals for 48 hours or for 10 days. Just initially, we've done 6 animals in total to try and understand, one, if we get expression, delivery, and looking at the safety. And our new R01 is now looking at the safety and efficacy of this across a much larger cohort of animals. So in the non-human primate placenta, by RNA scope, looking for the Texas red and the transgene expression, we can identify that we do indeed get expression of that transgene. The nanoparticle delivers this to the trophoblast. It's present at 48 hours, and in fact, we still see some signal out at 10 days. So again, looking very good for the idea of not having to treat maybe even weekly, but maybe able to push that out to treatments every 2 weeks as we develop towards the human. Is the transgene functional? So when we deliver IGF-1, we see changes in IGF-1 signaling within that primate placenta. So yes, when we deliver that transgene, it's not only present and expressed, but it is indeed functional as well within those cells and causing what we would see as normal function at both the 48 and 10-day time points. So we've gone on now to do assessment of both the placenta and mum. We've had the placentas analyzed by the non-human primate pathologists in Wisconsin, completely independent, and we see no increase in the occurrence of histological lesions at either of those time points. Once again, we see no evidence of immune infiltration by histology and CD45 immunohistochemistry, nor do we see any maternal immune response to the particle itself. Again, it looks like that polymer is doing its job. And at 48 hours, we see a change in the glucose transporter. So again, that would be indicating that you'd be increasing glucose transfer to and across that placenta. And mum's chemistry, again, looks very normal. These are not models of growth restriction. There are very poor models of growth restriction in the non-human primate, as the primate placenta can actually compensate and overcome the growth restriction by the end of pregnancy. But as far as safety and efficacy for delivery and function of this, we're on our way, and we're very excited to go on and do approximately another 25 animals over the next 3 years. To see if we can really push this actually through to the human clinic. So in summary, we've developed this intervention using trophoblast-specific therapy. It's able to be used after establishment of FGR, and so that's perfectly fitting for the human. The delivery so far across all of the species that we've looked at has been safe, effective. We have seen no off-target effects in any maternal or fetal organs, and it doesn't cross that placenta. It appears to work mechanistically by not only improving placental development and structure, but also changing the signaling and the function of the placenta and the syncytial trophoblast. There are fetal sex differences, which we will have to be very aware of as we move forward. People may be aware that male neonates are at more risk of problems within the NICU. And so if we can treat male fetuses in a different manner or better, that may be of an advantage. But the therapy is effective in both fetal sexes. And we see the same effect in the normal non-human primate as we've seen in the control guinea pigs. And so the potential for targeting placental therapy, of course, is not limited to fetal growth restriction. And that's what we're hoping to bring to this, this research project moving forward, is to start broadening the types of complications in pregnancy that we're interested in. And of course, this particle isn't only useful for actually therapeutics, but could also be used to disrupt aspects within placental development to form new models of complications as well. So as they say, it takes a village, and we have a big village that stretches across the country. I'm really grateful for all of our collaborators. This is a really exciting project, and the fact that we've gone on from just understanding what's disturbed in these placentas to actually now considering being able to actually directly treat them and really change the lives of somewhere between 10 and 20% of the humans on this planet. So I'll take any questions.
Thank you, Helen. That was amazing. Yeah, any questions? Let's see. Meghan. I don't know who was faster, but Meghan first.
I wasn't ready, but okay.
Sorry.
Super exciting, Helen. I mean, I've been following this for years, and I think it's amazing. One question I've never got the chance to ask you. So because you're using a polymer and you're loading it with plasmids, is your polymer restricted to negatively charged substances, or can it be combined with different things? Or is it mostly designed for like DNA, RNA delivery?
So the thing about the polymer is we can change its chemistry. And so potentially we could deliver anything. It doesn't have to depend on charge. The nice thing about using charge is the fact that once it goes into that Clathrin pit, it releases. And so there's no way it can actually go beyond the first cell it encounters. So it's a sort of safety level as well. So it's useful for that kind of delivery and safety mechanism as well. And for us, because we want to do plasmid delivery now, there could be all sorts of nucleic acids in that plasmid. So you could have RNA, you could have any of the small RNAs that you might want to disrupt something. You're not really limited to just DNA.
Thanks.
There's one question in the chat for you. Merlo Graton says, congrats to both of you. Dr. Jones, do you know if the IGF-1 nanoparticle deposits in other maternal tissues besides the placenta?
So it does go to the other maternal organs. We've actually now got ear injection. You can't inject a tail in a guinea pig because guinea pigs don't have tails. So we have to inject their ears to do circulatory delivery. And we do see it in other organs, but it's turned over very rapidly within about 72 hours. And because of the specificity of the promoter, there's no off-target expression at all. So even if the particle gets there, it doesn't actually express. And so it's then turned over by the cell within about 72 hours.
Okay. All right. And then the second question he had was if these particles have been tested in other fetal growth restriction models, such as RUP or fetal hypoxia.
So we've done surgical intervention with a specific hypoxia from the uterine artery branch. We've done genetic models in the mouse, and we're moving on to look at a couple of models in the non-human primate that we might consider. We haven't done the RUP because I have spent the last 20 years of my career trying to avoid preeclampsia, and now I'm not allowed to anymore. So.
All right, thank you. There was another question, but I don't know if it got removed. I don't see it anymore. Sorry. Please ask it again if. Yes. So Mariela Chertov, sorry if I didn't pronounce that correctly, is asking you, thank you for the excellent talks. Maybe I lost it, but how long does the expression of IGF maintain in the placental cells?
So from what we can see in vivo, we can see expression 10 days after delivery. In vitro, if we use some of the cell lines, which we can actually get the trophoblast to survive for more than the 6 days of syncytial culture, we also see about 10 days. We are going to do some longer term with the non-human primate in the current project to see if it goes beyond that. But it seems 8 to 10 days seems to be it starts to drop off quite quickly at 10 days.
Yeah, I think I may have missed it, but how often do you give the injection was it just one injection in the guinea pig?
So in the guinea pig, in the mid-pregnancy, we just gave one, but in the one that actually fixed the growth, we gave one every 8 days.
Okay, all right, yeah.
And we always say with that caveat, that might seem a lot, unless, of course, you're a heavily pregnant woman that's already at high risk and going in to see your clinician twice a week, in which case once every 8 days isn't very often.
That's true. Very good point. Okay. And did you see any, if I can ask a question, I think there's no other questions. Did you see any sex differences also in like the gross measures, like the placental weight or fetal weights, besides the mechanisms?
So we don't see them in the gross placental weight or fetal weight, but we've also taken all of the fetal organs for the guinea pigs. And so we do see differences in fetal brain and fetal heart with sex as well. So sexual dimorphism is a huge issue that everybody needs to be addressing.
And then have you, or are you planning to look at the offspring like later in life? I don't know if there's anything known about this model in guinea pigs and what the impacts are later in life. And
Yeah, so this model has been used huge amounts for the later life. Our co-PI, Tim Renaud, uses this model to look at non-alcoholic fatty liver disease in later life. But my junior faculty member, Beck Wilson, now, her K99I00 grant is actually looking at the offspring, looking at the impact on the liver, the kidney, and the reproductive organs within the offspring.
Awesome. That's exciting. Any more questions? Have you seen any change? This is again Mariela Chertov. Have you seen any changes on long-lasting effects in the FGR, such as brain function and cognition?
So we haven't done those experiments yet. They're in the next grant for the guinea pig. We've looked at blood-brain barrier development and all the different cell types within the brain. And the differences that we do see in FGR are ameliorated with the placental treatment, but that's only at term. We don't know about long-term going on in life, but we assume if your brain looks normal at term, then chances are it's probably going to stay normal. So
I think one maybe take-home message as well was kind of saw a similar thing as what we have been seeing with our nanoparticle delivery in prenatal hypoxia, is that you don't really want to give it in normal pregnancies.
well, we can
thoughts on that.
That's one of the things we can do. We get the beautiful homeostasis in the placenta if we give it in the normal pregnancies. So there's actually no problem with giving ours in the normal pregnancies. And in fact, FGR is overdiagnosed by a lot of obstetricians. And so it's actually extremely important that it's not going to do anything in the normal. And so the placenta seems to turn on its homeostatic mechanisms and it doesn't respond. It realizes it's got lots of IGF-1 and it just turns it down. So we saw no overgrowth in our treated controls at all. So.
All right, if there's no more questions, then yeah, I would like to thank both of you, Meghan and Helen, for these amazing talks and for a successful session here again. Again, Michelle will be here to answer any questions and the presenters as well if you want to stay on. Yeah, we really want to thank you all and remind you all that we are interested in the pregnancy research, your pregnancy research. So if you also want to present here, just contact the email address from WCHRI in the that's in the chat, and talk to Michelle about participating in the research cluster. Anything to add to that, Michelle?
No, thank you so much. It was absolutely exciting to have you guys in this space, and we really appreciate your presentations today.
Thank you all, and thanks to everyone for attending.